Hello, and I'm happy that I can do for you next lecture. As you see, the topic will be two parts. The first part will be the development of the urinary system, and in the second half we will speak about the development of the testes, of the ovaries, and all other things around this. Before we start with embryology, I think that I should explain a few words about the basic organs and basic ducts and passages which we have to develop. As you see on this simple anatomical scheme, so we are speaking about the development of the kidney, of course the urinary passages, that means some passages inside the kidney still, intrarenal, and the extrarenal passages. As you see, from the pelvic part of the kidney is coming out the ureter. The ureter is opening into the urinary bladder, and from the urinary bladder is coming out the urethra. So it's not only the problem of the kidney to develop, we have to also to speak about these parts of the urinary passages and the urinary bladder and the urethra. Here is a simple picture of the kidney. You see the capsule, little bit opened, the capsule from the surface of the kidney. This is the suprarenal gland. This is not our topic today. And we see on the section through the kidney the cortex. This is this part, very important cortex, and here this pyramidal shaped part which we call the medulla. You see here the minor, small calyxes and the large calyxes, and here we are in the pelvic part of the kidney, and here this is coming out, the ureter. The second very important thing is the nephron, please. This is a basic functional and morphological unit in the kidney, a nephron. The nephron has four basic parts. The first part is the renal corpuscle, containing the glomerulus of capillaries and the Bowman's capsule. Then the proximal tubule. Then the loop of Henle, descending limb, ascending limb. Then the last and fourth part of the nephron, the distal tubule. So please, the nephron has four basic parts. Renal corpuscle, proximal tubule, loop of Henle, and distal tubule. These four parts are forming each nephron. In the gray color you see the collecting tubules, which are opening and collecting together the urine into those little bit bigger tubules, which are called ductuli papillares. So, collecting tubules and ductuli papillares are not anymore the part of a nephron. Not anymore. They are on the apex of the pyramid in this moment, and here is a minor calyx, the space of the minor calyx. So the collecting system of the urinary passages are the calyxes, renal pelvis, ureter, urinary bladder, and the urethra. So this was a very short anatomical and histological intro to our embryology. So please, now if we know little bit, then we have to develop those very important urinary passages. We have to come to the urinary passages inside the kidney, and we have to develop nephrons in the kidney. So On the beginning, we have to start with our simple schemes or simple photographs, a section through the embryo. So here you see the scheme, and let's very quickly repeat what you should know already from the last winter semester. So here you see the closed neural tube. Under the neural tube you see the developed chorda dorsalis, notochord, the two dorsal aortas, and the closest mesoderm, if you remember, the closest mesoderm to the neural tube and the notochord is the paraaxial mesoderm. Paraaxial mesoderm. This is a cross section through the notochord. This is the closed neural tube, and this is the paraaxial mesoderm. As you see on both sides, in a short distance from the neural tube is the next part, next type of a mesoderm, and in this moment we speak about the intermediate mesoderm, and this is our very important intermediate mesoderm for the development of the urinary system and the testes and the ovaries. So the last, please, if you remember, the last mesoderm on the side here is so-called the lateral mesoderm, as you see on the scheme in two such plates. Yes. So this is the most important part, intermediate mesoderm, for our today's topic lecture. Intermediate mesoderm. Those cells are forming such a cord along the body of the embryo, which is called the nephrogenic blastema. Nephrogenic blastema. So this is our intermediate mesoderm. You see it, and now in this intermediate mesoderm, or in this blastema, we are slowly and steadily developing the urinary system. The urinary system in a human is not developing immediately, definitely, into a kidney, but you see that there are such slightly overlapping systems which are developing in one direction from the cranial portion, from the cranial part, to the caudal part, in this direction. Three important systems. The first system, here you have the time period. The time period is very short, very short. As you see, it's very high in the cervical region, very high in the cervical region. And this pronephros, the first system, pronephros, are such segmental cell clusters, which are called nephrotons. And there is also formation of a very simple duct, which is called the pronephric duct, as I can show you on the picture. Yes, so on a cross section, yes, please, a closed neural tube, the notochord, dorsal artery, dorsal aorta. If you remember the segmentation of the paraaxial mesoderm, so you have there somites, yes, somites. Even here is a small cavity. So this is the problem of the paraaxial mesoderm. And now we are in our famous intermediate mesoderm, because already this is the lateral mesoderm, as you see, so we need this. And those 7 to 10 units very cranially are forming nephrotons of this pronephros, solid cell groups, and here would be a small cavity of this pronephric duct. So the first system is pronephros, developing for a very short time. It will completely vanish. For a human, it's nothing important for the definitive kidney, it's nothing important for the genital system. So this is such a first system developing very early, very cranially, and will completely vanish, as you see on this picture. So this is a pronephric system here, cranially, and here were the clusters of cells and nephrotomes, and this short duct, violet, as you see here red, is the pronephric duct. Pronephric duct. There are also other types of nephros. As you can see, there is also existing hollow nephros or opistonephros. Such systems are with the pronephros, with this system, developing in some types of animals, in some types of fishes. It's present in amphibians, and so on. So here we see nicely the first system which will completely vanish, the pronephros, with the pronephric duct. And here you see it. So this is a tube, the digestive tube developing. You see the liver. Yes, so I think it's clear. And now we can move slowly and steadily to the second system. Which is starting around the 4th week, and the name is mesonephros. Mesonephros. You see that in this moment a duct on this side will be very solid and will be very long, and you see here it's in a blue color, and this duct is ductus mesonephricus, the mesonephric duct, in another name the Wolffian duct. Wolffian duct. Mesonephric duct, in another name Wolffian duct. And how develops the mesonephros? The mesonephros, as you see, from the 4th week, and slightly functional to the end of the 10th week. That means there are nephrons in each segment which are opening into the mesonephric, or in another name Wolffian duct. You see that we are not so cranially anymore. We are coming to the segment L4. So, and now you see the tubules of mesonephros. They are prolonging. Even you see here a simple glomerulus. And now this tubule of mesonephros, how is prolonging? Is changing the shape from a straight shape into a S-shaped loop, as we see on this picture, S-shaped loop. And on the end, it's opening into the Wolffian duct, mesonephric duct. On the end, it's opening into the mesonephric duct. So, and now we have here a very important ridge in this moment, which we see on this section here, on this scheme, which is called urogenital ridge, from here till here, with this part urinary ridge, and medially, that means closer to the dorsal mesentery, is so-called the genital ridge. So, plica urogenitalis, urogenital ridge, is seen at this moment with two subtypes, the genital ridge and the urinary ridge. Here you see once more how this S-shaped loop of the channel or tubule of mesonephros is opening into the Wolffian duct. And here is a small glomerulus. A very simple Baumann's capsule. So you see the function can be here in this moment. The function of this system can be. Please, to the paramesonephric duct we will come later. This is not so important in this moment. So, on the longitudinal section you see the system of mesonephros, and the Wolffian duct is here. Mesonephric duct. Mesonephric duct. Fine. And now we can come slowly to the last system, metanephros. So mesonephros was explained, and you see that the last system is developing from the 5th week, coming into the function around the 10th week. You see in which region? Lumbal part. And we see here a metanephrogenic blastema. One very important thing in this moment from metanephros, very important structure. And a very second important structure is the ureteric bud. I will show you in few seconds. So now we are slowly developing, if you read already those structures, so we are slowly coming to the definitive kidney. When you say metanephros, we are really developing the definitive kidney. This system has vanished. This system is vanishing slowly, and we will need only some tubules of mesonephros at the male reproductive system, because we need some important tubules in the epididymis. So, and we are moving to the second part here, that at the female completely disappears, and at a male you will see pictures in few minutes when we are developing the testis and the epididymis, that we will need few around 12 to 20 of those excretory tubules of mesonephros, between 12 to 20, in the place where we will develop the head of the epididymis, and in the head of the epididymis are present the passages which are called ductuli efferentes. Ductuli efferentes in the head of epididymis at man has the origin in the tubules of mesonephros, please. In the tubules of mesonephros. You see the dorsal mesentery, and this is the urogenital ridge, and the second urogenital ridge you see on this place. The same, the dorsal mesentery, and here is the urogenital ridge on both sides. And we are at the last system, metanephros. In this region you see how is developing this very important structure, the metanephrogenic blastema. This. You see that the mesonephric tissue with the tubules will be slowly vanishing and disappears, except at males, if you remember, this ductuli efferentes in the head of the epididymis. Here you see the Wolffian duct. You see here. And the Wolffian duct is opening into the cloaca. So what we have still here in the posterior part of the body. So we have the ending of the hindgut, as you see, the space of the cloaca, the allantois, which is running as this short process into the developing umbilical cord, in the connecting stalk, later umbilical cord, allantois. And we see here our famous similar membrane, which is on the opposite side of the embryo. So the similar membrane, the cloacal membrane. In the cranial part is the oropharyngeal membrane, and here you see the cloacal membrane. So, and a new structure is in this moment here. From the Wolffian duct, just before the Wolffian duct opens into the cloaca, rises this diverticle, and this yellow diverticle is a ureteric bud. And it's growing out from the Wolffian duct and penetrates the metanephrogenic blastema. Grows inside, in the metanephrogenic blastema. So this is a very second important structure to develop the tubules in the kidney. So this is a metanephrogenic blastema, ureteric bud, Wolffian duct, mesonephric duct, Hink gut, cloaca, cloacal membrane. And now the ureteric bud. Now the very important ureteric bud. What will be from this structure? So logical. From this structure will be the whole ureter. As you see, the pelvic part of the kidney, pelvis, the large, major calyxes, the minor calyxes, and we have to go on in the division, and ductuli papillares, and collecting tubules, please. And collecting tubules. So please, those all passages are from the ureteric bud. The whole ureter, pelvic part of the kidney, large major calyxes, minor calyxes, ductuli papillares, and collecting tubules. This everything is from the ureteric bud, please. And the epithelium of the collecting tubules with these factors is a very important inductive structure, as you see, that has to happen the transformation in the metanephrogenic blastema. Has to happen a transformation into an epithelium, because now is still missing all four parts of the nephron, and the nephrons will develop from those metanephric tissue caps, from this. So the collecting tubules are the real inducting structures for the development of the nephron. You see it slowly and steadily developing in these four schemes, how the nephron is prolonging, and of course we are developing the Bowman's capsule with the visceral list, which are special cells which are sitting on the capillaries of the glomerulus, and we call those cells podocytes. Podocytes will form the visceral list of the Bowman's capsule. The parietalist of the Bowman's capsule is a simple squamous epithelium, Between those two lists is the urinary space where will be running out the filtrate from the blood, the primary urine. Then the primary urine runs into the proximal tubule, simple cuboidal epithelium. Then the urine runs into the loop of Handler. The thin part is lined with a simple squamous epithelium. The thick part of the loop is lined with a simple cuboidal epithelium. And you see that we have to develop the fourth and last part of the nephron. And this is a distal tubule lined with a simple cuboidal epithelium. So here you see by blue color, the blue color from the metanephrogenic blastema, the nephron. Collecting tubules, ductuli papillares, small calyxes, large calyxes, pelvis of the kidney, the whole ureter are from the ureteric butt. So you see here that the ureteric butt is giving a very important origin to those passages, and they are inducting the collecting tubules, and the cells of the collecting tubules are inducting the development of all four parts of the nephron in the metanephrogenic blastema. You see the tissue caps, above, here and here. Here is one, here's another one, above the epithelium of the collecting tubules, how they are dividing. So this is the lumen in the collecting tubules, and here are the cells of the metanephrogenic blastema, and they will slowly and steadily now transform into the epithelial tissue of the distal tubule, loop of Henle, proximal tubule, and the Bowman's capsule. This transformation you see slightly on this picture. This is also important to know that the kidneys are rising slightly cranially into the definitive position, where they should be. So you see the short ureteric butt, you see metanephrogenic blastema, you see the Wolffian duct, ductus mesonephricus, and you see the cloaca. So, and now the simple ascensus of the kidney coming in cranially in this direction, prolonging the ureter. You already feel the pelvic part of the kidney. Already here you see that the septum is dividing the cloaca into two parts. So the ascensus of the kidneys is thanks to the growth of the body. The curvature of the body is starting to be more straight. It's not so bended in this region, the body of the fetus, and logically also thanks to this ascensus there will be a new blood supply for the kidney from the aorta. The kidneys comes functionally around the 12th week. Really around the 12th week they come functionally. Of course the function to bring oxygen and the function to take out from the blood of the fetus the urea and the uric acid is still the very important function of the placenta, not of the kidney at this moment, during the fetal development. So, and we have moved to the cloaca. To the cloaca. A septum will grow out in the direction to the cloacal membrane. In the direction to the cloacal membrane will grow out a septum urorectal septum. When this septum will touch the cloacal membrane, in this moment we cannot have one cloacal membrane, but we have divided this bigger cloacal membrane into two smaller. So logically, One will be membrana urogenitalis, and the second membrane will be the membrana analis. Nevertheless, they will disappear around the 7th week. 3 weeks later, then is vanishing the oropharyngeal membrane in the cranial region. So around the 7th week, these two membranes will also vanish. So, and when we have formed the septum urorectale, the cloaca has divided into two parts. Ventrally is sinus urogenitalis, here, and dorsally canalis anorectal channel. This is this anorectal channel. So you see the cloaca has definitely vanished, and we have now two new parts here, sinus urogenitalis and canalis analis or anorectal canal. The membranes will vanish around the 7th week, completely disappears. So, I think you see it here, septum urorectale, this is this, vanished the membranes, you see dorsally anorectal channel, and this is sinus urogenitalis. I think you feel the ureteric butt developing into the ureter. Yeah? Developing into the ureter. Yes. So, the sinus urogenitalis has now three parts. This upper, we can call it the vesical part, will give origin, as you see, to the urinary bladder, because the ureters are opening into the urinary bladder, left, right, ureter. So this is very clear, and now the connection is called urachus to the allantois, this connection. The lumen should completely vanish, and it will change into ligamentum umbilical medianum ligament. Ligamentum umbilicale medium. Sometimes can happen that the lumen is staying in the center. Then you can see the urachal fistula, or you can see a cyst, or you can see even a sinus urachi in the center of the urachus. The middle part, someone is calling the pelvic part, will give origin to pars prostatica and diaphragmatica or membranous part of the male urethra. Female, the whole urethra of this part. And now the last phallic part of sinus urogenitalis will give origin to the spongious part of the urethra and to the vestibule of the vagina. Don't forget that we've been in the cloaca. If you remember this picture, we've been in the cloaca. So that means hingut and cloaca and allantois are lined with endoderm cells. That means in this moment that the lining, the transitional epithelium in the urinary bladder and in the next passages, the origin is from endoderm. The urinary bladder is lined with the transitional epithelium, urothelium, transitional epithelium, and here in this moment the origin is from endoderm. Like the epithelium in pars prostatica and diaphragmatica and spongiosa of the urethra. So please, the sinus urogenitalis, sinus urogenitalis has three parts. So the vesical part, the pelvic part, and the phallic part. So, and I think in our second part we can move slowly to the development of the genital system in this moment. But once more, we haven't got it still now on the practical courses, so we have to speak a little bit about the basic structures on the beginning, at a male, at a female. The difference now is very important. So, as you see, the most important organ is the testis, with the very dense connective tissue on the surface, tunica albuginea, with a double list of peritoneum, because the testes are moving down through the inguinal channel into the scrotum, taking with a double list of peritoneum. We see lobules inside with seminiferous tubules. So this is a factory where is happening the production of the spermatozoa. So, and on the surface is tunica albuginea. You see the tunica albuginea is thicker in the dorsal part, where we can see such spaces, open spaces, lined with a simple cuboidal epithelium, which is called rete testis. And now we move from the testis into the epididymis. Epididymis has three basic parts. This part is called the head. This part is called the body, corpus, body. And this part is called the tail of the epididymis. You see, I have been speaking already about the tubules of Mesonethras, and you see them here now, the definitive result at a male, ductuli efferentes, which are taking the spermatozoa, and the spermatozoa flows from rete testis into ductuli efferentes, and those 12 to 20 tubules at a human are connecting together, forming a very, very convoluted long duct here, ductus epididymidis. Ductus epididymidis is partly in the head, but it's only in the body and in the tail of the epididymis. Carrying on, as you see this duct, and this is ductus deferens, which is running in the spermatic cord. Ductus deferens runs in the spermatic up to the urinary bladder, around the urinary bladder, and opening. The last part of ductus deferens is called ductus ejaculatorius, and this duct is opening into pars prostatica urethrae, into the urethra, in the male urethra, in the prostatic part of the urethra. When I say prostatic part, the urethra is surrounded by a system of prostatic glands at this place. So, ductuli efferentes, ductus epididymidis, ductus deferens, and ductus ejaculatorius, and the next passage at a male is still the urethra piece. The pars prostatica, pars diaphragmatica, and pars spongiosa of the urethra, up to fossa navicularis at last. So we have to come during our next part to the formation of the testes and to the male excretory, genital or reproductive passages. Female. So of course we have to speak about ovaries. Ovary, left side, right side, ovaries. Then we have to also to develop uterine tube, tuba uterina, left, right. You see the fimbriae, this is the ostium abdominale, this is the ampullary part of the uterine tube, this is the isthmus of the uterine tube, and this is the pars uterina in the wall of the uterus. Then you see that when the uterine tube is ending, we are in the cavity of the uterus with fundus, body, corpus, isthmus is this part, and the cervix of the uterus, cervix uteri, cervix of the uterus. And then you see here a little bit opened the wall of the vagina, So it was very simple and very shortly explained, little bit of the basic anatomical organs and structures, but I think it's very important to know it before we start to develop them. So, if you remember, I was speaking already about the genital plicas. Those ridges are medially, if you remember from mesonephros, you see, if you remember this scheme, because this is the dorsal mesentery, so here is a genital ridge. These are the S-shaped tubules of Mesonethras, and this is Wolffian duct. And here is the ductus mesonephricus. So closer to the dorsal mesentery, on the medial part of the urogenital ridge is the genital ridge. Here you see it, and here you see it. Yes, very nicely, and here. Yes. So now you see the proliferation. On the surface is the proliferation of the coelomic epithelium here, and under is the mesenchyme, and under the coelomic epithelium is the mesenchyme. Yes, so you see it on this moment. Here is the surface, coelomic epithelium, and here is the mesenchyme under. Yes. And to this place has to come the primordial germ cells. Now has to happen the migration of the primordial cells into this genital ridge. So, if you remember gastrulation, starting at the 3rd week, formation of ectoderm, mesoderm, and entoderm. The epiblast cells, I'm just repeating the most basic information, that the cells of epiblast cells are giving origin to all 3 germ lists. The first developed list is entoderm, please. So this is happening in the 3rd week. Now the yolk sac, forgotten L here, yolk sac. So the first primordial cells are seen or can be distinguished among entoderm cells in the wall of the yolk sac around the 4th week. 1 week later, those cells are starting migration along the dorsal mesenterium, here, you see it, and they are starting to occupy on both sides the famous genital ridges. So this is the migration of the primordial germ cells from the yolk sac in the wall of the dorsal mesenterium and occupying on both sides the genital ridges. This is lasting around 1 week. So around the 6th week, the first cells are arriving to these both ridges. They have to reach, because if they are not coming to this place, those very important cells from the yolk sac, the testes and the ovary will not develop. So this is very important. On this picture you see the same, how the primordial germ cells are moving and occupying the genital ridges on both sides of the dorsal mesenterium. On both sides of the dorsal mesenterium. So very important, they have to reach this place and this place for the development of the testes or the ovaries. So, and here you see the coelomic epithelium on the surface. Yes. And now the cells from the epithelium penetrates inside the mesenchyme. You see the primordial germ cells arriving, the red, and the same situation would be on the opposite side here, please. Yes. So they are occupying and arriving into the genital ridge. And now the first cords from the surface epithelium coming down into the mesenchyme, and these first cords we call the primitive sex cords. At this moment, you cannot tell till now if this will be a testis or if this will be an ovary, please. At this moment, not. This you know already, this is a simple glomerulus, this is a Bowman's capsule, and this is an S-shaped loop of the tubule of Mesonephros, which is opening into the Wolffian duct. Wolffian duct. So this was at the urinary system, and we will need mesonephric duct, Wolffian duct, also for the male genital system. Invagination of the coelomic cells at this region, invagination of the coelomic cells are forming a duct at this place, and this is para, means very near, para-mesonephric duct, in another name Müllerian duct. Müllerian duct is a para-mesonephric duct at this place. So, at a female will completely vanish. At a male, if you remember, few of those tubules will give origin to ductuli efferentes in the head of the epididymis. And now, at a male, it's clear that this chromosome, one gene is present, sex-determining region, and if it's present, this gene on this chromosome, epsilon, the development will go into the testes. At girls, at ladies, when it's missing chromosome epsilon, then this gene is not present, and the development will go into the ovary. So, you see how it's important to the ridge, this is ridge, and you see coelomic epithelium on the surface. We've been speaking about the first cords, and now there are some factors of those coelomic cells which will go on in the development right into the testis. You see here the Wolffian duct, very important for male, and not important the Müllerian duct at male. The paramesonephric duct will disappear. Opposite. The mesonephric duct at female not important. But very important, the Müllerian duct, the paramesonephric duct. The paramesonephric duct, very important. Of course, the right development into Sertoli cells, pre-Sertoli cells and later Sertoli cells, very important cells in the seminiferous tubules. Leydig cells, very important for the production of the hormone, of the steroid hormone, testosterone, please, testosterone. And seminiferous tubules with the later from the puberty developing the spermatozoa. Opposite here, the oogonia, germ cells, will be surrounded by follicular cells, forming the first primordial follicles, with the oocyte in the center and flat follicular cells in one layer around this oocyte, and we will have primordial follicles. So, I think you understand the tubule of mesonephros, the Wolffian duct, yes, the Müllerian duct, but for a male, not important. And here the surface celomic epithelium running inside the cells, forming the primitive sex cords, and now we see new cords, which are called medullary cords from the epithelium. They will be so horseshoe-shaped, later seen, and the cells will completely get away from the surface, and this connective tissue forming here is forming slowly and steadily tunica albuginea. So the tunica albuginea separates these horseshoe-shaped medullary cords, which will be the basis for the seminiferous tubules later, from the surface epithelium. So here you see nicely the horseshoe-shaped medullary cords in the developing testis now. Some tubules remained of mesonephros, that you feel the head of the epididymis, and of course our famous ductuli efferentes, which you have seen a few minutes ago. And they are opening into the Wolffian duct, ductus mesonephricus, yes, which will form, I can say to you already, ductus epididymidis and ductus deferens. If you remember, the paramesonephric duct at a male will completely vanish. A male hasn't got the uterine tube, hasn't got the uterus. So the paramesonephric duct will vanish. So, Leydig cells will differentiate from mesenchyme cells, because Leydig cells are not lying in the seminiferous tubules. Leydig cells are lying in the loose connective interstitial tissue around the seminiferous tubules. So the Leydig cells are differentiating from the mesenchyme cells, in the developing testis. You see nicely the surface celomic epithelium. Here will be tunica albuginea. This, and you see the medullary cords. Nicely in the developing testis. Ovary. So, the medullary cords were present. And now they are starting to disappear, as you see here. Medullary cords were present. But they will completely disappear, because now from the 7th week starting a new second generation of cords coming out the cells from the surface epithelium, and the new cells forming so-called new cortical cords forms around the 4th month such clusters around the orgonia, and you have the primordial follicles on the world. So this is the problem of the developing secondary generation, the second generation of cords. Cortical cords, as you see on the first picture, cortical cords. And later, those epithelial cells from the cellum will surround the orgonia in one layer, and we will call them follicular cells, around intensively division. You can see divisions of orgonia, intensive mitotic divisions of orgonia. So the number of primordial cells and of primordial follicles is rising. And here are vanishing the former medullary chords, but at a female, not at a male. You see that the tubules of mesonephros we don't need at a female, because a female hasn't got epididymis. So they have to completely vanish. And now we'll start the regression of the Wolffian duct, of ductus mesonephricus. But the Müllerian duct is now in this moment very important at a female. So you see that this will vanish, medullary chords will vanish, Wolffian duct for nothing. This is now important at a female. Müllerian duct or ductus paramesonephricus. Paramesonephricus duct, Müllerian duct. So, at a male, Wolffian duct. Ductus epididymidis, forming the body and the cauda of the epididymis, the whole ductus deferens running up in the spermatic cord, and the last part, ductus ejaculatorius. That's the problem of the Wolffian duct. The Müllerian duct completely degenerate. The rest is known as appendix testis. You can see it sometimes above the epididymis. So, Sertoli cells are present in the seminiferous tubules. Very important cells for the development of the spermatozoa. There are many Sertoli cells in each seminiferous tubules. They have many functions Sertoli cells, and they are also producing factors, if you want, hormones. And one of those factors is AMH, so-called anti-Müllerian hormone. In some books you can find the name anti-Müllerian factor, and this inhibiting substance produced by Sertoli cells suppress the Müllerian duct, because the male hasn't got the uterine tube and the male hasn't got the uterus. Testosterone. Leydig cells, if you remember, differentiate from mesenchyme cells. So, very clear now the development of the external genitalia. No. Uterine tube, uterus, and upper portion of vagina. That's the problem of the paramesonephric Müllerian duct. Yes. Estrogens, which are produced by the cells of theca folliculi interna. This is a layer around the follicle, and with the help of follicular cells are produced steroid hormone estrogen, or some type of an estrogen, and these estrogens are stimulating this. So, you feel the developing seminiferous tubules. Seminiferous tubules, tunica albuginea on the surface, yes, surrounding the seminiferous tubules, and the mediastinum testis with the spaces rete testis. Then you remember our famous ductuli efferentes, the tubules of mesonephros, as you see here, forming the head of the epididymis. The body and the tail is the problem of Wolffian duct, ductus mesonephricus, the whole ductus deferens of the Wolffian duct, and the last part before opening into the urethra, ductus ejaculatorius. And here, if you remember, I was speaking about the appendix testis. At a male, the rest, a small rest of the Müllerian duct is seen as appendix testis, such as small diverticle above the epididymis. So here were very important at a male, some tubules of mesonephros. At a female, completely disappears. Mesonephros, completely disappears. The testes which are developing in the body cavity has to move in the channel to the scrotum. This is called the descensus of the testes. It's lasting around 3 months, starting around the 5th month, the descensus of the fetal development. On the end of the 8th month, one testis is jumping into the scrotum. Few days later, the second testis should enter the scrotum, and they are lying in the scrotum. The reason? The ideal temperature, please. The ideal temperature is clear because the right temperature for the development of spermatozoa is around 35.4 degrees of Celsius. This is the right temperature which should be in the scrotum, because in the body cavity there is a higher temperature. If the testis, one or second, will get stuck in the way to the scrotum and will not enter the scrotum, will not arrive inside, then the situation is called cryptorchism. Cryptorchism, it can be on one side, one testis, unilateral, or it can be on both sides, both testes, and this is bilateral cryptorchismus bilateralis. Very unpleasant situation, because when they are in the body cavity or in the channel, the higher temperature is not an optimum temperature for the development of the spermatozoa. So this is the whole problem why the testes has to come down into the scrotum, the ideal temperature for the development of the spermatozoa. So you see how the developing ovaries, yes, the vanishing medullary chords and developing cortical chords, the next generation in the developing ovary, and you see the follicles already here. And now the tubules of mesonephros has to vanish because the lady hasn't got epididymis. And the lady's female hasn't got ductus epididymitis, hasn't got ductus deferens, so this everything has to vanish. The rest of the mesonephric duct you can see here in the mesovarium, epithelial cells, such clusters of epithelial cells, which is called ophoron and paraophoron. So this can be the rest of the Wolffian duct at a female. Now, how is important the Müllerian duct, paramesonephricus? So you see that the cranial part and the horizontal part will form the definitive uterine tube. Will form the definitive now uterine tube, tuba uterina, uterine tube, with the ostium abdominale and those long processes, very important during ovulation, which are searching the Graafian follicle just before the ovulation. The ampullary part of the uterine tube, where happens the fertilization of the oocyte at a human. The isthmus of the uterine tube and the pars uterina of the uterine tube, in the wall of the uterus. This last part of the Müllerian duct is forming the uterine channel, or in another name, canalis uterovaginalis, the base of the corpus of the uterus, as you feel, and the cervix of the uterus, and a small part of the vagina. That's the reason why it's called canalis uterovaginalis. But here you see the uterine channel. So, this has to vanish at a female. This is important, the Müllerian duct, for the development of the uterine tube, the uterus, and a small part of the vagina. So the vagina, you see here the septum, because this was the left and right Müllerian duct. So the septum will vanish and you will have the cavity of the uterus. Here, opening of the left and right uterine tube. Here this is the caudal tip of the Müllerian duct, So you see a small piece of the vagina is still from the caudal tip of the Müllerian duct. And now the rest. These cells of the sino-vaginal bulb, they are growing caudally, forming a longer vaginal plate. And in this vaginal plate, you can see first cavities, connecting cavities. So it's completely canalized around the 5th month. And here is the ready vagina. So the vagina has two origins. A small part is from the tip of the Müllerian ducts, and the rest is from the so-called sino-vaginal bulb, forming a vaginal plate. And you see how it's canalized around the 5th month. So, on the beginning of the external genitalia, you see the indifferent stage on the beginning, where we can see the genital tubercle or tuberculum genitale in Latin, and two cloacal folds, here and here. The cloacal membrane, of course, is still seen up to the, if you remember, 7th week, then it vanishes. The cloacal folds will divide into two parts, the urethral folds and here the anal folds. So there are two parts of the cloacal folds, two parts. And when we see new elevations, So we see genital swellings on both sides of the urethral folds, a new elevation here on both sides. As you see, this genital swelling will give origin to the labium majus, here and here. The genital tubercle is giving origin to the clitoris and opening of the vagina and the urethra, and the labium minus are seen on both sides from the urethral folds. The phallus, the genital tubercle at a male phallus, is prolonging, and the two urethral folds will fuse in the middle line that you see here, the line of fusion of the urethral folds, and forming the nice part of the penis. Of course, also the urethra, penile urethra will close, and you see then the lumen in the center. So this was very shortly now explain the external genitalia. So, my dear friends, thank you for maybe reading or listening to this lecture about the development of the urinary and genital system.